Okay, so what I'd like to start with today is actually a story that started, well, I try and think of like how old I am, so decades ago now. So back when I was in graduate school, transgenic and knockout technology in rodents was just being developed, and so I went to graduate school at UT Southwestern in Dallas, and one of the researchers in that area, Rob Hammer, was a leader in the field of making transgenic animals. And so as an immunologist, one of the things we had learned about was that in looking at disease associations is that one of the strongest genetic disease associations was, and actually I would probably argue even as much as we know about GWAS and SNPs and stuff, it still remains a very strong disease association between a genetic phenotype in humans and disease is the expression of HLA-B27. And so to do Koch's postulate cause and effect testing, the group there made transgenic rats in which they expressed the human HLA-B27 gene in rats. And what they were able to find is that these transgenic rats expressing HLA-B27, and then they also, because HLA-B27 is in, it's a dimeric molecule, or I guess it's a heteromeric molecule. So it's in association with beta-2 microglobulin. So they transgenically introduced both of these genes into rats, and what they found is that these rats would develop a spontaneous inflammatory disease that involved the gastrointestinal tract, the peripheral vertebral joints, male genital tract, the skin, nails, heart. So in other words, reproducing many of the features that you see in the human autoimmune diseases that are associated with this gene. Now, interesting side note that no one's ever talked about is they actually had made transgenic mice to start with, and they didn't develop disease. And so that's why they gave up on them, because they went to the rats. They figured something wrong with mice, you know. But this story moves forward then, because you move forward a couple years, and what they found is if they took these rats and they put them under germ-free conditions, they re-derived them so they had no microbiome in them, they did not develop the inflammatory disease of the intestine or peripheral joints, but they still got some of the other diseases, such as the skin and the genital inflammatory diseases. And so that should have been the wake-up call at that point in time, that the microbiome can really play an important role in immune modulation. But it was a very slow process, and so these studies were now beginning to, so other studies started to appear after that from other laboratories, again supporting this concept of potentially the microbiome in immune regulation. But what I really want to also emphasize, if we go back a second, is that these animals, these subjects, are genetically predisposed to a disease. Okay, if I can say nothing else, if you remember nothing else from my talk today, okay, this is a gene that has a very strong association with disease. Okay, yet you change the microbiome, they can be protected. Okay, so when we get to this idea that's been talked about at this symposium already of a gene-environment interaction, okay, there are many diseases, I'm going to argue, that we think have genetic associations that are actually gene microbiome associations. Okay, and so you can change that, and again, gene microbiome environment associations. So as an immunologist, the simplest way to view immune regulation is it's really about a balance. It's a balance between inflammatory processes and the processes of control inflammation, so anti-inflammatory processes. These exist at the level of adaptive immunity, at the level of innate immunity. And so one of the things we've been interested in is pursuing this idea that perhaps the microbiome plays a role in this balance. Okay, so the idea that there could be certain microbes, or more importantly, what I would argue, community structures of microbes that may favor maintenance of an anti-inflammatory state, so in other words, a balancing state, or those that can promote inflammatory states. And so the idea is, conceptually, is that if you, in an individual, a switch between a microbiome that would favor strong anti-inflammatory responses, which would give you very good immune regulation, to one which you could favor development of inflammation, that concept is dysbiosis. Okay, so a switch in the microbiome towards favoring disease or degradation of health. And so now, in terms of allergies and respiratory diseases, if you go through the literature, this little chart, which I don't have any intention of you reading, all I expect you to do is look at the number of lines that are on there. This is a chart that we made for a review article probably about 5 or 6 years ago, and this shows a number of studies that had come out at that point looking at an epidemiologic association between fecal microbiota composition and allergies. In other words, differences, like in individuals that are allergic, are there differences that we could detect in the microbiome? And this is prior, all this data is prior to the advent, to the use of high throughput technologies and the use of culture independent microbiology to study this. And so if we switch our associations and say, well, what about antibiotics? Okay, so not necessarily the microbiome, but actually use of a significant microbiome perturber such as antibiotic use. Again, a large number of studies that were showing associations. And I was kind of, as I was sitting here through the presentations this morning, I was trying to think, what kind of context can I put these sort of studies in? And what occurred to me is that the way that our studies in the microbiome and its role in physiology and health, it's a lot like looking up at the sky at night. Okay, so if you think about primitive cultures, you look up at the sky at night, okay, you see some stars, you don't know exactly what they are, they're little blips of light, but then there's a lot of black space between them. Okay. As we developed technologically, we developed telescopes. You start to be able to look up into the sky. You see that in that black area, there's actually more stars. Okay, and as our technology develops more and more, as you can then stretch your imagination, you realize that some of those blips of lights aren't stars. They're actually they can be galaxies, and within that, then the things you could have had a hard time seeing, which are planets. And then as you get closer and closer, you realize that when you have a planet, that there may be life forms on that planet. And as you get closer, then you say, well, why don't all the life forms work together? And it's like, oh, it's because there's bodies of waters that separate them geographically. And then as you get closer, you say, well, what about the ones that are all in the same body? And you say, well, they speak different languages. And as you get even closer to that, you say then they have different cultures. So my point that I'm trying to get to is the fact that there's a lot that we haven't known because we haven't been able to see it. And the closer we look at the microbiome, the more amazing it's becoming, because it's my analogy of like looking at Earth. So if you were on the opposite side and you were out in the distant galaxy looking at Earth, what would you really know about us? Okay, so it's not until you spend and you get close and you realize how we interact to understand how all this works. And so from that level, then, I look at these old studies as this is the first, these studies here that are looking at these associations are the first telescopes that we had to say there's got to be something going on, because these associations are not strong, they're not always consistent, but there's something there.
Sorry. So I was just curious about this correlation, whether or not they had canvassed people and said, what was your antibiotic usage in the past? And do you now have allergies? I mean, how was this?
Yes, that's exactly how, in general, that's how all these individual studies were done. And some of the studies were done, they could actually go down and look at different types of antibiotics, so whether it's a broad spectrum or narrow spectrum, and others were just antibiotic use. Always the confounding factors are, for something like asthma or allergies, were you taking the antibiotic because you already had health problems, or did the antibiotics precede the health problem? And so again, the studies were getting there, but that's what they were looking at. So one of the things that our laboratory did, and I won't go into the story of how we actually got to this stage, but using this idea of antibiotics, that there's a possibility that perturbation of the microbiome could alter allergic diseases in the airways, what we set up to do was actually to test this in a mouse. And as simple as this experiment is to do, it had not been done, which either makes me lucky or insightful. You take your pick. And so what we did is we set up an animal model. We did this in both inbred black 6 mice and inbred BALB/c mice, so we had some genetic variation in it. And we treated the mice for a week, so 5 to 7 days with a broad spectrum antibiotic. So it was cefepozone, so it's a 3rd generation cephalosporin, and then we challenged them in the airway intranasally, either with mold spores or with a known test allergen, so ovalbumin ova. And what this data shows, the groups themselves are microbiota disruption. So this is using the antibiotics to disrupt the microbiome, or they are challenged intranasally with the mold spores, or it's a combination of the two. Remember, these are genetically immunocompetent animals. Okay, so these are, in a sense, healthy animals walking around the cage. And what you can see is the big yellow bars. And so what is that? So we looked at, in the airways, we looked at IL-5 production, IL-13 production, the number of eosinophils coming in, and even the number of mast cells. And what we could see is that if we treat these animals with an antibiotic, oh, and by the way, this is after they've been treated. They're treated 5 days of antibiotic and then let go. Okay, so they are not on the antibiotic. This is post-antibiotic. This is 2 weeks post-antibiotics. And you can see they start to develop an allergic phenotype. Actually, it's a very striking phenotype. And this is kind of my histology for dummies slide, which is if you look at, so I think it's on your left here. Right? Anyhow, you look at this here. So this group over here. So that is your normal airway. And this is a stain looking for mucus production. And you can see the open airways. You see that there's not much in the way of mucus, a few goblet cells that are making mucus. But in animals that had been previously treated with the antibiotics and now challenged with the allergens, indeed they made a significant amount of mucus. And so we changed the whole phenotype of this animal without changing its genetics by changing its microbiome. And so now we went on to show that this is a CD4 T cell dependent phenomenon. It's IL-13 dependent. Didn't matter about the 2 different genetic backgrounds we looked at, and didn't matter about whether it was ovalbumin or Aspergillus mold spores. The other thing we've done since then is we've actually looked at whether this antibiotic, is it antibiotic actually restricting its activity to the gastrointestinal tract? And indeed it is, because it's a poorly absorbed antibiotic. It's given in the water. We've actually been able to put cefepozone susceptible organisms in the lungs of mice, put them on the antibiotic water, and indeed it doesn't touch the lung itself. So this is a distal site phenomena. And by the way, this antibiotic is normally given intravenously because it's so poorly absorbed from the GI tract. So again, it emphasized that, where's most of the microbiome? It's in the GI tract, right? And so the question is how would this potentially work? So the model is, the theory is an idea of a gut-lung axis of immunoregulation. And so you typically think of immune responses happening in the lungs. You inhale an antigen, so such as an allergen. You develop your response in the lungs. But we also have to keep in mind that virtually everything we inhale, we also swallow. So especially particulates. Particulates are going to get caught in the mucus layers in the upper airway. They're going to get kicked up. They're going to get swallowed. And there's been a number of studies that have shown that actually when you do intranasal challenges with human beings or mice, it is not just staying in the nose and it's not just going into the lungs. It is always being swallowed. Always, always, always. You can't when you're looking at that. And so if that's the case, then that means that whenever you have an antigen that's going in the lungs, it's also going into the gut. And that gut is under, the immune response in the gut is under the influence of the microbiota in that area. And so, as we'll be talking about later in the afternoon, what are the various metabolites that the microbiota might be making? But there's two different things. So one is the fact that the gut immune system can circulate. Okay, so whether it be regulatory cells, whether it be other factors. So the idea that we may be developing basically immunizing and tolerizing at two different sites in the mucosa, and the mucosa is talking that way at an immunologic level, but also the idea that there may be, not maybe, I'm almost positive there are metabolites that are being made by the gut microbiome that are circulating, that are affecting also the lung environment. Now, this is, again, I go back to this picture because we think about where all the bugs are. Okay, they're from the mouth all the way down to the GI tract. But the mucosa, okay, which is where the bugs are found, remember there's a cul-de-sac in that mucosa. It's called the lungs. Okay? And so if you look in textbooks, the idea is the lung is sterile. Okay, so there is no microbiome in the lungs. You get exposed, a few bugs are quickly destroyed, and it's only when you're sick that you really have you have any bacteria in the lungs. And so the question is, one of the questions is it possible that this model that I have up here is indeed correct, but it also that the microbiome of the lungs may be playing a role. Okay, so I told you in our animal model that is not likely the case in that particular situation, but that doesn't necessarily mean it's true in all situations. And so one of the interesting things is to answer that question about are the lungs sterile, when the NIH set up the Human Microbiome Project, it was decided not even to address that question. Okay, so the initial round of the Human Microbiome Project, if you notice where they have all their sample sites, you notice that the lungs are not on there. And so subsequent projects, especially pushed by National Heart-Lung Blood Institute, has decided to look at some of those things, although not directly, kind of indirectly, so looking at it in a disease setting. But one of the things that we've worked on recently is actually asking the question in the human lung, is it indeed sterile, using culture-independent microbiology techniques. Now, how do we get this idea that the lungs are actually sterile? It's because if you take a healthy human lungs, it is difficult to consistently culture microbes out. Okay, in fact, in some studies it would say 0. And so when we looked at this using culture-independent techniques, what we found is that in every single sample we looked at by bronchoalveolar lavage, there's indeed bacteria in there. So bacterial signatures, so 16S signatures. And so this, we looked at healthy smokers, patients that have COPD at various degrees of intensity, and non-smokers, and indeed everybody is coming up positive. When we looked at it in terms of its composition, at a phylum level, I'm sorry, this is actually down at a genus level. The first thing you'll see is that, it calls our beach ball analysis, is that there are some individuals that have very almost monolithic, so very limited diversity in their lung microbiome, and others that have quite a bit of diversity. We don't know what that means. We also don't know if the microbiome that we're seeing in the lungs is a stable thing, or if it's a whole series of transients that are coming and going. We know nothing about that. But one interesting thing we did is we also took lungs out that were transplant lungs. Okay, so these are patients that had really severe COPD. They're having their lungs transplanted, so the lungs that were coming out. What we did is we sampled all around different sites and actually asked questions about local anatomy of the microbiome. And what I want to point out is if you look in the upper corners here, so in CS number 6, you'll see 3 of those beach balls there. One's mostly red, one's mostly purple, one's mostly light blue. What those are is those are domination of sites that are geographically or physically really close to each other in the lungs. One's dominated by Pseudomonas, one's dominated by Haemophilus, and one's dominated by Stentrophomonas. But then if you look at other sites in the lungs, you see a little bit more diversity. So there's a heterogeneity within the lungs. Now, granted, this isn't a disease state. We haven't looked at like trauma lungs or things like that. But we're just beginning now to scrape the surface of what is the lung microbiome and what's it mean in terms of health and disease. We're not the only ones that are looking at this. As I mentioned, there are a number of NIH-funded projects, but a couple other studies I want to point out. This one came out in PLOS One just prior to ours, and the conclusion from this study was that the bronchial tree is not sterile. The results show the bronchial tree to contain a characteristic microbiota and suggest this microbiota is disturbed in asthmatic airways. Another study came out the conclusion was that phylochip analysis revealed the presence of over 1,200 bacterial taxa representing 140 distinct families, many previously undetected in airway diseases. And then I go back to this because we've actually known that, like I said, in disease states such as cystic fibrosis, there is a microbiota there. There is a bacterial communities there. And so in this particular case, this is looking at cystic fibrosis. And so they used, again, culture-independent technology, and they revealed that enormous complexity of bacteria within the cystic fibrotic lung. And so the idea is not just one or two bugs in CF. There's actually many there. And so my final couple slides I want to come back, return to, is this idea then of the environment and its impact on the microbiota. And so if we look at just a handful of things that we do potentially to the microbiota, so between antibiotics, our diet, so we've talked about carbohydrates. I mean, it's clear that the different types of carbohydrates will represent different signals, perturbations, feeding to the microbiome. So do you have a diet that's high in simple sugars? Do you have one that's high in resistant starches? That will have a significant impact on the microbiota. And then obviously, if you do something like eat bacteria, like probiotics, you could change the microbiota. But also many medications, non-steroidal anti-inflammatory drugs, have been shown to alter the microbiota. What about protein pump inhibitors? And you go on from there. And so the idea and then in the diet, the phenolic content of the diet, it can play an important role in modulating the microbiome. And so now I return to the original story I started with. Okay, so back to our HLA-B27 rats. And so again, remember that these guys develop an inflammatory disease. Okay, but here's an interesting study that came out in 2009. They took these rats and they actually gave them lyophilized apples from two different cultivars, so the Golden Delicious or the Marie Magnard. And the main difference that they were interested in testing was these two different types of cultivars have low polyphenol content or high polyphenol content. And what they could actually find is that that one of the aspects of disease that these rats get, in this case it was the colitis, they could actually ameliorate it when the rats were fed a diet high in polyphenols. And so again, come back to this idea then that one of the ways that we can positively modulate the microbiome is again by what we eat. And such that diets that are high in your colorful raw fruits and vegetables, which have high phenolic content, may play a positive role in reshaping the microbiota. And so on that note, I'd like to thank my sponsors who've kept me and my lab alive for quite a while. And then this is my current group, with a special thanks to my collaborator Vince Young, who's been my microbial ecology mentor in all of this. So thank you.